in and so my topic is to talk about biological rhythms and their modeling and the importance of limit cycles in cellular regulatory systems. And thank you for the organizers for the invitation. This, I think, is the second time I come to Le Havre. First time I do. I have some memories that it was a while ago. So, before entering, oops, I have to see how to progress. Yes. Before entering into the modeling part, I would just like to stress the fact that rhythms occur at all levels of biological organizations with widely different periods, ranging from less than a second in neural rhythms or in the cardiac rhythm, the order of 1 second, to minutes or even years. So, as you can see here, we have calcium oscillations which occur in many cell types with periods of seconds to minutes. There are oscillations in some biochemical systems. I will come back to that. There are hormonal rhythms. Most hormones are released periodically in pulses. The mitotic oscillator is driving the cell cycle. I will come back to that. Circadian rhythms occur in all eukaryotic organisms with periods about 24 hours, and I will also discuss that topic. Now, all these rhythms are marked by an asterisk. It means that they can occur already at the level of single cells. But sometimes they occur because of ring interactions between cells in networks, like in neural networks, for example, where you have both the possibility of oscillatory behavior in single cells and oscillatory behavior resulting from interactions in a neural network. And then you have above the cellular level, you have the ovarian cycle, you have annual rhythms and rhythms in ecology and epidemiology with periods of years. Now, all these rhythms have been approached experimentally, but also theoretically. And in the last 20 years or so, additional rhythms have been uncovered. One of the most fascinating is the segmentation clock, which underlies the formation of somites in vertebrate embryos. The period of the phenomenon is of the order of 30 minutes to a few hours, depending on the species. You have a transcription factor NF-kappa B, which oscillates a tumor repressor P53, a transcription factor in yeast called MSM2, which goes into and out of the nucleus with a period of a few minutes when the cell is under stress. Then you have the whole yeast transcriptome, but most importantly, I would like to stress the last line, the appearance of synthetic oscillators. And the first example was devised in 2000, actually, by Leibler and Elovitz. Elovitz and Leibler published a paper in Nature about a synthetic gene regulatory network, which they studied experimentally and also theoretically. And they called it the repressilator because it consists of 3 repressors coupled cyclically. A inhibits B, B inhibits C, C inhibits A. By the way, this pattern is also known in neurobiology. And there are papers long ago which stress that this organization can give rise to oscillations. So all these rhythms have been modeled theoretically, most of them at least, especially when the mechanism is known. But probably one of the first works which dealt with modeling is due to Volterra. And the context, as you know, is ecology. Volterra wrote a famous paper in 1926 where he considered predator-prey interactions and showed that these interactions can give rise to oscillations. And actually, this is the scheme. It's a scheme where X is the prey and Y is the predator, and there are 2 kinetic equations which are not highly nonlinear. They are nonlinear, but not very much so. And as you know, when you integrate these equations, you get oscillations in time. This is just a sketch that I took from one of the lectures that I gave. So it's by hand. It's an approximation, as you can understand. And then if you plot in the phase plane predator population versus the prey, you have closed curves, but you have an infinity of closed curves. Depending on the initial condition, you have the amplitude varies as well as the period. Now, this type of oscillation has to be distinguished from the one I'm going to discuss at length in this presentation. And it remains a central topic in ecology, in population biology, where you see here, that's on the cover.
Okay, j'arrive.
By Alan Hastings. There is a strange voice coming there. Now, another field where oscillations are extremely important, I already mentioned that, is neurobiology. And there is a beautiful book by the French neurobiologist Alfred Fessard, which was published in 1936 by Hermann, the publisher. And the book is entitled Propriétés rythmiques de la matière vivante, Rhythmic Properties of Living Matter. But the subtitle is These Are Isolated Nerves, because Fessard was mostly working with nerve cells, and this is an example, later of course, of an oscillation of the membrane potential in a nerve cell, and in the neuron R15 in aplysia, which is really as famous in neurobiology as Escherichia coli is in microbiology, you have bursting behavior, and this is another form of complex periodic oscillations, which is well studied in neurobiology. So, of course, the theoretical work has been devoted to neural oscillations. There are many names of researchers who are extremely active in the field. And I think if John Rinzel is listening to this talk, because he was earlier on there, John, of course, is one of the key figures in modeling neural rhythms. But it began with the work of Hodgkin and Huxley, the theoretical work. And as you know, Hodgkin and Huxley got the Nobel Prize in Physiology or Medicine in 1963 for their discoveries of the ionic mechanism underlying the action potential. And one of the 4 papers they published in 1952 in the Journal of Physiology was devoted to the modeling of the action potential. And so this is the founding work, and their model continues to be used in modeling the oscillatory behavior and the excitable behavior of nerve cells. Because this model, to get an oscillation, you have to supplement it by a current, a depolarizing current, and that was also shown in subsequent papers. Now, I will not talk about this type of oscillation. I will talk about biochemical oscillations and the role they play in cellular rhythms. And I will start with an example of an oscillation which still remains the prototype of oscillatory behavior in a biochemical system. And this is, it's called glycolytic oscillations. They are observed in yeast cells or yeast extracts. It was the first example of a biochemical clock observed in vitro. And the mechanism was discovered early on, around 1964. And if you inject constantly sugar in a suspension of yeast cells or in yeast extracts, the sugar is transformed into CO2 and ethanol. But it is transformed in a periodic manner. Although the input is constant, the output is periodic. And the period of the phenomenon is of the order of a few minutes. Now, it's an interesting system because you can identify a control parameter. The control parameter is the input of substrate injection. Here you see that 40 millimolar per hour, and if you go to 20, you change the waveform and the period, You go to 80, you change the waveform in a period. And if you go too low, 20 here, you go to, you lose the oscillations. And they resume if you increase again. So there is a domain of oscillatory behavior. And this is the reason why it remains a very interesting experimental system to work with. And as I said, the mechanism was uncovered early on. Glycolysis is a chain of enzyme reactions. And it was shown that one of the enzymes of the pathway plays a key role. It's called phosphofructokinase, PFK. PFK, and why does it generate oscillations? Because it is regulated in a peculiar manner. It is activated by a reaction product. So this is the scheme of a model which was proposed long ago with René Lefebvre, and it depicts a system, an enzyme reaction, where you have an input of substrate S, which is binding to the two states of an enzyme, PFK, an active state and a less active state, transformed into product P, which is removed. And P activates the transition from less active to more active. So it's a kind of self-amplifying process, a positive feedback. And so, of course, so how to model that? You can try to model the whole pathway, or you can try to focus on this enzyme reaction and just retain the key regulatory feature. And this was done with René Lefebvre in a paper which dates back a long time ago. So you can see from that picture that I worked on oscillations for quite a while. But I remain fascinated by the topic. And the word allosteric is important. It means that the enzyme, it's because it is a source of cooperativity. So when you look for oscillations, you realize early on that you need nonlinearity. And there are many sources of nonlinearity. One source is the feedback process, activation by the product. The other is the cooperativity.
What does allosteric mean?
Allosteric means that another form. The enzyme exists in two conformations. There was a beautiful picture for that given by an enzymologist of the name of Kasper Kirchner. He said, think of an umbrella. An umbrella exists in a conformation. If there is wind, strong winds, it switches to another conformation, right? So you don't have, actually it's called concerted. And so Monod, Wyman, and Changeux proposed a model for the concerted transition between the two states in 1965, in a classical paper published in the Journal of Molecular Biology. So this model is based on the Monod, Wyman, Changeux model, but it incorporates the positive feedback. Alpha is the normalized substrate concentration, gamma is the normalized product concentration. And V is the input of substrate, phi is the rate function of the enzyme, this is the removal of product. The rate function phi is highly nonlinear. You see, this is the positive feedback term here, and you see that it's non-polynomial, and L is called the allosteric constant. So this model accounts for many of the experimental observations. It can evolve to a stable steady state, but it can also produce sustained oscillations. This is sustained oscillations in time, where the substrate alpha oscillates here, and the product gamma oscillates in a highly pulsatile manner. And if you plot the substrate versus the product in the phase plane, then this curve was generated at the time with an analog computer, which people do not use anymore. But it's beautiful because the curve is produced very slowly, and you can see the trajectory evolving to a closed curve, the thick black line, and you can also reach it from inside, from the unstable steady state. You rotate until you reach the curve, which is called the limit cycle. Okay? So the stable limit cycle can reach both from inside and from outside. And now, because it is a two-variable system, I insist on this because I will discuss more complex models to try to contrast the use of simple models versus complex models. The advantage of a simple model containing two variables is that you can use phase plane analysis. And in this case, the phase plane analysis, there are two curves which play a key role. They are called the null clines. This is d alpha over dt equals 0, okay? D alpha over dt equals 0, and d gamma over dt equals 0. This equation here with phi given by this curve. And the intersection is the steady state, because it is the only point where at the same time you have alpha dot equals 0, gamma dot equals 0. Now, you can apply a linear stability analysis and translate it into the phase plane, and you find that the instability condition is that the slope d alpha over dt, d alpha over d gamma, so the tension here at the steady state, that's a subscript 0, has to be negative, less than minus 1 over q. Q is a parameter which appears here. And the steady state is simply equal, of the product, is simply equal to QV over K. Okay. V is the input of substrate. So if you increase the input of substrate, you go from left to right. The steady state goes here, 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 here. Now you can follow, here the steady state is stable, the slope is positive. But here, if you are here, the slope becomes negative. It becomes unstable if it is sufficiently negative. And then if you continue, the slope ceases to be sufficiently negative, and then it's stable again, okay? So this phase field analysis explains why, so this is an illustration, the null cline d alpha over dt equals 0 for 3 values of v, 0.1, 0.6, 1.8, stable steady state, unstable steady state, and this is the limit cycle. So, so this is the key experimental observation, which is accounted for this analysis, by this analysis. If you increase V, you have at first a stable steady state, then you reach a critical value, sustained oscillations occur, then you reach an upper critical value, a stable steady state is reached again. Now, more complex phenomena can occur if you coupled in series two instability-generating reactions. What do I mean by that? Here, you recognize here the model, the schematized for the PFK, you recognize here, you recognize the PFK model, okay? One positive feedback loop. What happens if P1, the product, is the substrate of an enzyme, E2, a second enzyme, giving product P2, which would activate E2? What happens when you couple two positive feedback loops in series? A positive feedback loop, you have seen, generates an instability and oscillations. And Ks is the rate constant for the removal of product P2. Now, this work was done with Olivier de Crolly a long time ago, and by doing that, it's a three-variable system. So now we have three ordinary differential equations with two rate functions of the kind of the one we saw, and we can generate chaotic behavior in this model. That was originally the idea, to look for a simple model for chaos. And then one day, Olivier de Crolly came in and said, I have got a result which is strange. If I integrate the equations for two different initial conditions, I do not get the same oscillatory solution. In one case, you see that's the first oscillation, and in this case, we have that. Actually, we found also conditions in which there were three coexisting stable solutions. This is an unstable solution. So this is a case where of birhythmicity, coexistence between two stable periodic regimes. It is the counterpart of the well-known phenomenon of bistability, where you have a coexistence between two stable steady states. Bistability plays an important role in different cell differentiation processes. That's only one example. And, but this phenomenon of birhythmicity has not yet been investigated experimentally in detail, and there are very few examples. So, and if you vary the parameter Ks, the rate constant for the removal of product P2, you see that you go from, you can observe periodic oscillations. This is the substrate as a function of time. You can obtain here chaotic dynamics, and here below you can obtain bursting behavior. So either you evolve then to a strange attractor in the case of chaotic dynamics, and below you have bursting oscillations. Now I will turn to other systems that we studied later, which pertain to two of the most important cellular regulatory networks. The first example is the cell cycle, and the second example is the circadian clock. At the end, I will discuss the coupling between these two oscillatory systems. And I will use these two systems to discuss the use of simple models versus complex models. What are the advantages of simple versus complex? Because complex models are useful in other respects. And in discussing the dynamics of the cell cycle, I will start with the simpler case, the frog embryo, and then I will move to mammalian cells. So in the frog embryo, the first 12 divisions, the first 12 cell divisions occur regularly at 30 minutes intervals. And this was a phenomenon which allowed the discovery of the molecular basis of the cell cycle reuse. And as you can imagine, the cell cycle is one of the most important cellular processes because it is involved in development, going from the fertilized egg to the adult organism, but also it plays a role in cancer, because in cancer you have the uncontrolled division and the proliferation of cells. So this summarizes the knowledge at the end of the year of the 1980s. And it was found at that time that in the cell cycle, in the frog embryo, there are two phases: M phase, which is mitosis, cell division, and then interphase. And they follow each other every 30 minutes. And what happens is that during the interphase, there is a protein which increases, and then it decreases during mitosis. And at the same time, there is an activation, a transient activation of a factor called MPF. MPF is mitosis promoting factor. So that was established experimentally in the 1980s. But at the end of this decade, it was realized that MPF is a complex between a kinase. A kinase is an enzyme which adds a phosphate group to a protein. And thereby, it can activate the protein. In some other cases, it inhibits the protein. It's called covalent modification. And what happens here is that MPF is activated, and this leads to the degradation of cyclin. And because MPF is a complex, cyclin plus a kinase called CDC2. CDC is cell division cycle. It's the name given by two mutants of the cell division cycle in yeast. Then MPF is inactivated, and then it oscillates again, another cycle. So for the discovery of that phenomenon, three scientists received a Nobel Prize. Paul Nurse for this work on CDC2 in yeast, Tim Hunt for his discovery of cyclin, and Lee Hartwell for the discovery of mutants of the yeast division cycle. So how to model that? Now, in 1991, nearly at the same time, working independently, John Tyson, who is a good friend of mine for a very long time, published a paper in PNAS modeling the cell division cycle. And I insisting on the role of positive feedback. And nearly at the same time, a few months later, I published another paper in the same journal based on negative feedback. And so we discovered that this is the model which I show schematically. And it's interesting that I will compare that to the more complex model later. So it's the three-variable system again, where you have a synthesis of cyclin. Sorry, I will show it here. You have the synthesis of cyclin and the degradation of cyclin. Cyclin activates the kinase CDC2, by activating the dephosphorylation of the enzyme, and then there is rephosphorylation. And then this CDC2, when it is active, activates a protease X, which degrades cyclin. So this is a negative feedback, because when cyclin accumulates, it leads to activation of M, which leads to activation of X, which inactivates cyclin. Okay, so these are the three equations. Just Michaelis-Menten kinetics, but strongly coupled variables. Okay, because here V1 contains so V1 here, where is V1? V1 is here. Activation of M is triggered by C, and here V3 here, activation of X is triggered by M. And if you integrate these equations numerically, you see that cyclin increases linearly because it is synthesized at a constant rate. It triggers the activation of M, which triggers the activation of the protease X, the dashed line, which leads to cyclin degradation. And then, because X goes down, and M goes down, cyclin can accumulate again. And so this is a very robust oscillation. Actually, the curves here were obtained in the first numerical simulations that I tried. So it is extremely robust. And if you plot one of the three variables, cyclin, for example, as a function of M, the fraction of active CDC2 kinase, once again, you see that the system evolves to a limit cycle, starting either from inside or from outside. Now, in the mammalian cell cycle, the situation is more complex, because there is a family of complexes between cyclin and what is called cyclin-dependent kinases. In the embryo, I mentioned CDC2 kinase, which is a kinase named CDC2, but in the mammalian cell cycle, there are different complexes, not just one. So, for example, and different complexes control the transition between different cell cycle phases. For example, the transition to mitosis. So there are two main phases, mitosis and S, which is DNA replication. And these M and S phases are separated by G1, gap phase 1, and G2, gap phase 2. And this is the complex controlling the entry into M. This is another complex here, cyclin D CDK4/5, controlling progression G1, and so on. So you want to account for the dynamics of this system, and it is driven by a growth factor which triggers a transition from a phase called G0 into the cell cycle, into G1. So it's a CDK network, cyclin-dependent kinase network. And so I had a very good and excellent PhD student called Claude Gérard, who engaged in the modeling of the whole system. There were models proposed for parts of the whole system. And this is how the system was built. First of all, here you have the cell cycle phases on the right, G1, G1S, S, G2, and G2M. And there are four complexes between cyclins and CDK. So you see the first one is cyclin D, CDK4/6, controlling the corresponding cell cycle phases on the right. Then cyclin E, CDK2, cyclin A, CDK2, and cyclin B, CDK1. And I will not discuss in detail all the regulations, but to give you an idea of the complexity of the regulatory structure, I will show you the main player and the main regulatory loops. E2F is a transcription factor. There is a break on the cell cycle dynamics called by a protein called PRB, the retinoblastoma protein. When there are mutations in this protein, it can lead to cancer cells. Then you have the growth factor. Oh, sorry, the growth factor is here. On the top left. It triggers the synthesis of cyclin, And then you have E2F, which activates cyclin expression. You have PRB, which inhibits, it's an antagonizer of the activation. Cyclin D activates the inactivation of PRB. Cyclin E CDK2 completes the inactivation of PRB because there are multiple phosphorylations. Then P53 is another control of the CDKs through the inhibitor P21. And then this shows you here how the system works. What is remarkable in the organization is that you have these 4 modules. Each module activates the next one, but each module inhibits the previous one. When the task is done, then it inhibits the previous. So this is to ensure that it's irreversible. When you pass through a cell cycle phase, you don't go back. You go to the next one, but you inhibit the previous ones. And at the end, you have additional interactions. So this is only a representation of the most important regulations. But this is a more detailed one. And there is no wonder that the model is described by 39 variables. So I discussed the 3-variable model. Now we have a 39-variable model. Usually mathematicians will say that's horrible because, how can you handle that? Analytically, I agree that there's not much hope, but of course, we can use numerical simulations. And what are the advantages of using a detailed model is that all these parameters correspond to biochemical identities. So if you want to ask, what happens if I have a mutation, for example, in the kinase which phosphorylates one of the intermediates, then I can put its activity to 0 or a reduced value, and then I can see the effect. If you do not have a detailed model, you cannot ask such a precise question. So if you, now there are 39 equations, and this is the result of the integration. The integration is that there are spontaneous oscillations in the CDK network, and they are ordered. First, you have a peak here in the black curve, cyclin D CDK46, which induces Z1 progression. Then the blue curve, SG2, is controlled. Then the green curve, which controls the transition to M. And mitosis is controlled by the red curve, which is cyclin A CDK2. No, the red one is cyclin B CDK1, excuse me. Cyclin B CDK1. CDK1 in this system corresponds to CDC2 in the 3-variable model. Okay. But there are additional CDKs. And if you integrate, if you project in the phase space the 39 variable dynamics as a function of 3 variables, you evolve once again to a limit cycle. So what is remarkable is that here we have a highly complex system with 39 variables, which has the dynamics reducing to the dynamics that we saw in the 3-variable system. And these oscillations are extremely robust. Now it's possible to reduce the complexity of the model for the mammalian cell cycle, and this was done with Claude Gérard, with whom we built a skeleton model. And the idea was to retain the regulatory structure. So we still have the 4 modules, the same modules, and the main interactions are there. Basically, one module activates the next one and inhibits the previous one. But of course, what we removed is all the biochemical details. We removed the biochemical details and we only retained the basic regulatory structure. Now we have 5 variables and 25 parameters. Before we had 39 variables and more than 100. And then,
Albert, 5 variables and 25 parameters.
Yeah.
How can you estimate those parameters? How can you control?
No, this is not a quantitative model. This is a model, an exploratory model. Right? In the previous talk, you want to explore the possibilities. You explore the parameter space, and you take reasonable values. Okay, you know that the dynamics of the system, the period of the cell cycle should be of the order of 16 hours to 20 hours, okay? So we do not obtain 2 years or, and so if you, these are the equations of the variable system, we make another assumption, which is that one of the variables remains nearly constant. You see that in the full model, cyclin D-CDK4/6 does not oscillate with a high amplitude. So we take a mean value and we keep it constant. But you see that the reduced variable model, reduced variable model gives the same phase relationship, the blue, then the green, then the red curve, right? And so this is, if one wants to study the other dynamic feature, that's a good model to use. Now, I remember that I attended a conference in China once, and I had the luck to be sitting next to Jim York, the famous mathematician who coined the word chaos in one of his papers. And he asked me what I was working on. And then I told him, well, I'm working on a model for the dynamics of the cyclin CDK network, and it was very long. And then he said to me, I give you 3 minutes to find a better title. Then I was motivated. I gave plenty of titles until I came to the cell cycle is a limit cycle. He said, that's it. And then we published a paper with that title.
As he did, Pedro 3 price card.
Yes, so he likes short titles, right? And so this is much shorter. We had some difficulty with the editor of the journal, who thought that it was a statement which was very strong, but okay, but I thank Jimmy York for this wonderful experience. Now I turn to the circadian clock system, and before coming back to by uniting the two. And the circadian clock, it's like with the cell cycle. In the cell cycle, we start with one situation, the embryo in frog, and then we went to the mammalian cell cycle. And here now is the circadian clock. We start with Drosophila. Why? Because that was the system in which most progress was done experimentally at the time, initially. And first, some words about the circadian clocks. Circadian clocks allow the adaptation to the environment. And I think that there is one N which is superfluous. I realize that now. So what Ashoff, who was one of the most important workers in the field of circadian clocks, wrote is, one of the most obvious adaptive features of life on Earth is the ability of almost all species to change their behavior on a daily or 24-hour basis. So it means that living organisms adapt to the alternation of night, and it had many roles, like the sleep-wake cycles, jet lag. So just a few paintings to show that this theme of day and night has fascinated men for a long time, and this is seen in some artist work. René Magritte, the Belgian painter, in this famous The Empire of Light, he painted some 20 paintings with this theme, but it's remarkable. Now, Antonio Segui, who came from Argentina, has also day and night, okay? It is a painting that's more recent. And then Escher also had a wonderful representation of day and night. You see that the birds in black merge to the birds in white on the right. So what do we know? Yeah, this is from my personal collection. Now, these bottles I bought in Paris some 20 years ago, and I do not intend to drink them, because it's probably not a very good wine, but just it's the value is in the symbol. So the study of circadian clocks at the molecular level started with this remarkable paper by Konopka and Benzer. Konopka was doing his PhD thesis at Caltech with Seymour Benzer, who was a famous neurobiologist, and he went to Benzer, letting him know that he would like to work on circadian clocks, and Drosophila was the organism on which most genetic studies on development were done for a century. So he embarked on this project, and they came up with a paper, which is a landmark paper, in 1971 in PNAS, where they identified one gene, which they called period gene. And in Drosophila, the wild type has a rhythm. There are two rhythms. One rhythm is locomotor activity, okay? And then they rest. They move, they rest. And this has a period of 20 hours, or 24 hours approximately. That's the black lines are movement and then rest, the thin lines. And there is one mutant that they isolated, an arrhythmic mutant, and then they isolated a short period mutant, 19 hours, and a long period mutant, 28 hours. So this stayed like that for 20 years, this result. And people worked on that, on the period gene, okay, to understand where the mechanism, what was the mechanism of the periodic oscillation. And the progress was made by the group of Rosbach at Brandeis University in Boston, who in 1994 studied the expression of the gene. And so they have here, this is the RNA. As you know, the gene is transcribed into RNA and then translated into protein. So the two curves, one for the eye, one for the brain, show that there is one peak in 24 hours. This is 24 hours. This was done in a light-dark cycle, but they show that in total darkness, it also oscillates. But now we know the phase with respect to the light-to-dark transition. The peak occurs in gene transcription occurs a few hours after the transition. And then the protein is shown here. It oscillates with a delay of a few hours. And they remarked that when the protein increases, the RNA decreases. And so they suggested that the protein per for period, the protein per exerted a negative feedback on the transcription of its own gene. So this is an example of autoregulatory negative feedback. Now, this mechanism is observed in most organisms studied so far. It's the core mechanism of circadian rhythms. It's more complex than that because there's not one gene. At the time, there is one focus on one gene. Now we know that at least there are 15 genes, if not more. But it's not a mechanism which is observed everywhere. For example, in cyanobacteria, it's another mechanism, but I don't have time to go into that. And as you may know, Rosbach received the Nobel Prize for his discoveries that he did with Jeff Hall, and also Michael Young from Rockefeller University shared the Nobel Prize because he found other proteins involved in the mechanism, which I will mention in a second. So I will briefly discuss three models of increasing complexity for circadian rhythms in Drosophila and mammals. The first model is based on negative feedback of the PER protein. At the time, that was in 1995, only the protein PER was known, and the effect of light was not known. I did not yet mention that the circadian clock is sensitive to light. That is the way it allows adaptation to the alternation of day and night. You can have entrainment by the light-dark cycle. I will come back to that in the next model. So this is a model where you have transcription of the PER gene in the nucleus. This is one cell, and this is the nucleus of the cell, which transcribed into PER mRNA, which is degraded. This is translated into protein. It was found by Rosbach that the protein PER is phosphorylated. What was the role of phosphorylation was unclear at that time, but they assumed that it was involved in the transport into the nucleus. So it's multiply phosphorylated. So the simplest way to make a model with multiple phosphorylation is to take two phosphorylation. Actually, there may be more. So when the protein is phosphorylated, it goes into the nucleus, where it feeds back negatively on transcription of its own gene. And this is the degradation rate. And so this system is governed by 5 differential equations. The first term here is the Hill function, which describes the cooperative inhibition, because Pn on the denominator is the nuclear form of the protein. So the more protein there is, the less the RNA, the less the gene is transcribed. And this is the Hill function with a Hill coefficient of 4. When you integrate these equations numerically, you obtain for a certain range of parameter values. M is the red curve for per mRNA. The total protein per is the dashed line in blue. And the other curves show the monophosphorylated, non-phosphorylated, and dephosphorylated form. And once again, if you project the dynamics onto a two-variable phase plane, per mRNA versus total per protein, you observe the evolution toward the limit cycle. I think that the advantage of working with limit cycles is that it's less boring than a steady state. Steady states are very important, of course, but a steady state is a horizontal line. A limit cycle brings movement, and there is an aesthetic part also in strange attractors, of course, for chaos. So now the model number 2. The question always arises: when do you need to modify a model? You have to modify a model when there are new experimental data which are obtained and which bring additional important information. In this case, what Michael Young showed is that there is another protein which is important, which is called TIM. TIM makes a complex with PER, and it is the nuclear per-TIM complex which inhibits the transcription of the per and TIM genes. Oops. And in addition, what Rosbach and others, 4 groups showed that at the same time. It was, I think, in 1998. That light controls the circadian clock in Drosophila by inducing the degradation of TIM. So when the light intensity increases, the degradation of the TIM protein increases. And this brings a mechanism for the entrainment of the circadian clock by the light-dark cycle. So to incorporate that into a model, we now have 10 variables for the model. And in red, this is the regulatory terms, the Hill functions. And light controls this parameter. And so now we can study the model in 3 different conditions: continuous darkness, continuous light, and a light-dark cycle. And to simulate a light-dark cycle, we take a square wave. We go from a low value of degradation of TIM in the dark phase to a high value in the light phase. And this is the period. So if you take an oscillation, this is in continuous darkness. So you have a constant value of the degradation rate, and the period is about 23 hours. If now you drive it with a light-dark cycle of 24 hours, the oscillation is untrained to 24 hours. Okay? This is the fundamental property of the circadian clock. And in Drosophila, in continuous light, you have a damped oscillation, as observed experimentally. Now, curiously, and we did not expect that, in this model we can obtain autonomous chaos. That is for constant parameter values. And the reason is that there are 2 instability mechanisms which are hidden in the system, because we have the 2 negative feedback loops on TIM and on PER. And then we also can observe the coexistence between 2 limit cycles, 1 small amplitude limit cycle and 1 large amplitude. Now, the mammalian system? 10 years later, there were much experimental progress on the mammalian circadian clock, where the oscillation is controlled by cells in the suprachiasmatic nuclei, in the hypothalamus. And these cells are really responsible for the origin of the circadian clock in mammals. And in this beautiful paper by the group of Okamura, it is accompanied by a movie, and I hope, yes, it will probably work. What they did was to link the PER protein to luciferase. and which produces bioluminescence. So what will we see is the suprachiasmatic nuclei. There are two nuclei which are flashing every 24 hours during 3 days. And you can find this movie in the paper published in Science in 2000. Yes. So you see, below are the traces. 24-hour period. Now, what is the mechanism in mammals? It is similar, not identical, but closely related to the one in Drosophila. You have, once again, here on the left, you have two genes which induce, it's called BMAL1 clock, which induce the expression of purr and cry. We do not have TIM, we have cry. And so, and purr and cry bind to the activators clock and BMAL1 to inhibit. So it's indirect negative feedback. So this is a model which contains 16 variables or 19 variables, depending on whether you include this part here, which is involving another gene called alpha. And there are other models which have been proposed by Herzl, Forger, Doyle, Kim and Forger. So they, this detailed model was one of the first detailed models proposed. And the equations, once again, no, sorry, this is not the good place where to put it. This is a slide which belongs to the cell cycle modeling. This is the main result. This model can generate oscillations in continuous light, continuous darkness, or in a light-dark cycle, allowing entrainment to occur. So, to end, I would like to discuss briefly the coupling between the cell cycle and the circadian clock. And the two have been shown to be interlinked by regulatory laws. I will discuss unidirectional versus bidirectional. Unidirectional coupling leads to entrainment. For example, if one of the parameters of the cell cycle is controlled by the circadian clock, the cell cycle will be driven by the circadian clock and will be entrained by it. So 24 hours. But more recently, it was shown that the coupling goes in the two directions. It's bidirectional. So this is, the first coupling was unidirectional, where you see that there is a kinase called WEE1, which inhibits CDC2 in the cell cycle. It's controlled by the circadian clock, and so this leads to entrainment. But more recently, it was shown that there are additional loops which link the two. For example, in this scheme, this is the circadian clock and this is the cell cycle. The BMAL1 in the circadian clock controls B1, but now we have another one here in red, cyclin BCDK1 here inhibits the synthesis of these actors of the circadian clock. And if, so this can lead to entrainment. For example, usually if the circadian clock, which is always 24 hours, is linked to the cell cycle, let's say 16 hours, if you couple bidirectionally, they will synchronize at an intermediate period, either 17 or 19, depends on the relative strength of coupling. But what is remarkable and totally counterintuitive is that we can obtain sometimes a bifurcation diagram like this one. So this is the synchronization period, and this is the autonomous period of the cell cycle. Oops. So sometimes we have here, this is one synchronization only, and this is another synchronization. But here, where you have the three dots here, you see that the bifurcation lines are multiple. And so for this value of the autonomous period, around 20 hours, for example, and this occurs for certain types of bidirectional coupling, you have three modes of synchronization. Either here, the synchronization is 23.74, this one is 30.89, and this one is 25.2. So we have a phenomenon of multi-synchronization.
It's a kind of phase synchronization, not identical synchronization, right?
What? Of?
Phase synchronization, phase synchronization.
Yes, but also the period is different. The period is different.
Yeah, the parents are different, yes.
Yeah. And so, if you study that in a diagram in parameter space, this is the synchronization period as a function of the coupling parameter of the cell cycle to the circadian clock, and this is the coupling of parameter of the circadian clock to the cell cycle. You see that there are three surfaces, one corresponding to the high period in yellow, intermediate period in violet, and then lower period in gray. And if you look at that in the phase space, you have either one limit cycle, or you have two limit cycles. I did not show you that for other values of the parameter, we have coexistence between two stable limit cycles, or you have a coexistence between three stable limit cycles. And that we call that multi-synchronization in work done with Jie Yan in the university, Suzhou University in China. Suzhou is between Shanghai and Nanjing. And so we did this work together. Now, this is just one example of multirhythmicity, and I discussed other examples of multirhythmicity during the presentation. So, to summarize, limit cycles in biological systems. Oscillations are ubiquitous at all levels of biological organization. I focused at the cellular level. And then what is the reason? It's regulation. Actually, why are rhythms so numerous in biology? Because biological systems are regulated, they are controlled. In the course of evolution, regulatory mechanisms evolved, and this allowed nonlinearities to appear, which favor the onset of oscillatory behavior. I discussed limit cycles in glycolytic oscillations, which was the first example studied at the molecular level in a biochemical context. Then I discussed the cell cycle, going from amphibian embryos to mammalian cells, then the circadian clock, going from Drosophila to mammals, and the coupling between the cell cycle and the circadian clock, which is a topic which has been studied more actively in recent years. And I discussed the interest of simple models versus complex models. I think that both have their advantages. And actually, it's a good approach to study both in parallel if you need to do so. Of course, there are simpler models than 2 and 3 variables. Some people can use normal forms, and then you lose any link with the underlying biology, biochemistry. But that's fine. This was done, for example, by Conover for the circadian clock, when he studied properties of the circadian clock as a function of temperature, for example. And these models, once they give limit cycle oscillations, if you increase the number of regulatory loops which are interacting with each other, you can get more complex phenomena like bursting oscillations, chaos, or even multiple limit cycles. So I think that I will stop here, and thank you for your attention.